Welcome to the Waterloo Institute for Health Informatics Research Seminar Series. We are resuming from the fall series, started in 2006, with this seminar series. And the seminar today will be given by Dr. Melanie Campbell. Before I introduce Dr. Campbell, I just want to make two announcements. The next seminar of this series will be held on Valentine's Day, February 14th. And it will be given by Dr. Ann Pittick. She's going to be talking on clarity in confusion. How can we prevent dementia? Sounds like a really interesting title, so I hope you will come back and join us on February the 14th. We also run another seminar series which is celebrating the university's 50th anniversary. It's entitled Why Not? And the kickoff speaker for that series will be on, which is on Wednesday, January the 24th, will be Dr. Jake Thiessen. He's the head of the School of Pharmacy as well as the new Health Sciences campus at the University of Waterloo. And he will be speaking on why not bring medications and their use out of the dark ages. So I think it will be a very entertaining talk as well. Melanie Campbell and I guess my paths have crossed a number of times on this campus. I know that she's been very active in teaching, not only at the university level, but she's very interested in engaging students at the lower levels, going out into the public school system. She is also very active as a researcher. She crosses from Optometry, the School of Optometry, and the Department of Physics and Astronomy. One of the things that, if you read her bio sketch, is that she was awarded the 2004 Rank Prize in Optoelectronics for research, and this is a quote, where an initial idea has been carried through to practical applications that have or will demonstrably benefit mankind. That's quite an award, so congratulations, Melanie, on that. She's going to be speaking today on improved views in fundus imaging, implications for diagnostics and therapeutics. So, Melanie, will you please begin your presentation?
Thank you, Shirley. It's good to be here. And I'm going to give very much an overview presentation today. And I know a number of you specializing in vision science will have seen a lot of this before. I'm hoping to be able to allude to current work and future work to keep you interested. And I know there'll be some people in the audience as well who may not have been exposed to this area. So I'm treading a difficult line to keep you all interested, and I just hope that I can be successful in that. So first of all, the work I will talk about isn't possible without a very large number of collaborators, a number of whom, hopefully most of whom are listed here, and I will try to highlight people when I get to their individual projects, and if I don't, my apologies in advance if I miss highlighting someone's work. So today I'd like to speak about imaging of the eye, and particularly I'm going to concentrate on confocal scanning laser ophthalmoscopy, and I will explain to you what that is. And again, apologies that I won't have time to, or I will only have time to allude to some of our work in optical coherence tomography. And I will also highlight polarization imaging. That's assuming that I have enough time at the end. And that imaging, of course, has obvious implications to diagnosis, but I would also like to talk to you a little bit about human imaging and animal models as it relates to therapy, and in particular I want to talk about one of our projects on two-photon therapy and imaging in animal models particularly. And those animal models then are used for various applications in disease, understanding disease, and looking at therapies in disease. The very exciting advancement in imaging that I want to talk about is adaptive optics and how that has improved our views of the fundus, and of course I want to emphasize clinical applications. So we have the eye, which has two optical components, the cornea in the front and the crystalline lens as the second optical component. So that light comes from a distant object, in this case through the two optical components, and is focused onto the back of the eye. And this is really a key fact for the work that I will be showing you, because our view of the back of the eye is very important. The fundus, the structures at the rear of the eye, tell us a lot about ocular disease and also about the health of the body as a whole. We're interested in the blood vessels, we're interested in the optic nerve head, we're interested in the actual neural cells that are at the back of the eye in the retina, and all of those views are influenced by the optics in front of the fundus. And so my research is directed at fundamentally understanding this optics, but the research I will talk about today is directed at improving the views of the back of the eye through manipulation of the optics in front. So the imaging that I will talk about initially is confocal scanning ophthalmoscopy. How many of you know what confocal scanning laser microscopy is? CSLM? A few people? Okay, so I'll start then from scratch, even describing what a confocal scanning laser microscope is, which is this part of the slide. We have a light source. That light source is focused onto the sample that you're interested in looking at. Light then is reflected or scattered back from the sample and directed via a beam splitter to another lens, which then focuses the light onto a pinhole. And a key element of a confocal scanning laser microscope is this pinhole, and I will explain why a little bit later. And then the light is actually detected after it goes through the pinhole. So a confocal scanning laser ophthalmoscope is identical in principle to a confocal scanning laser microscope, except that the light must go through the optics of the eye, both in the ingoing and outgoing path, rather than through a high-quality objective. And so that's going to limit the resolution and the quality of the images we see, and we would love to be able to replace the optics of people's eyes with high-quality objectives, and I will show you how we actually go about doing that. So what happens if you have poor optics? Well, here we have an image with fairly high contrast and a lot of fine detail. And if you have poor optics, then what happens is that image gets blurry. But I want to point out two things that happen, because these are the same things that happen to us in our imaging. The first is that you lose resolution. So if we pick an area with very fine detail here, in this image with blurry optics, you can't see those details. They disappear completely. The second thing I want to point out, though, is even for the largest features that you're looking at, the contrast in the image drops because of the poor optics. So not only does it affect fine detail and resolution, but it affects the contrast of all features. And so improving the optics is really important, especially in eyes which are elderly or have disease, because the features we want to look at the back of the eye are already low contrast. And this blurriness of the optics will lower that contrast still further. Okay, so we have a confocal scanning laser ophthalmoscope, so I want to take you through what a couple of those words mean, and in particular what scanning means. So here we have a little movie from the time Austin Rorda was in my lab, and it shows the laser beam scanning across the rear of the eye and then being descanned on its way back to the pinhole here and the photomultiplier tube, which detects the signal here. And so you can scan, in fact, raster scan in 2 dimensions the beam, I'm only showing one here, and bring it back on axis so that it still goes through this pinhole. And then we recreate the image by a timing system that tells us when it's at each point on the rear of the eye, and we can recreate our video rate images of the rear of the eye. The confocal imaging, we focus this laser beam down onto the focal plane of interest, and that's the solid line here, which is in focus on the plane. And that light comes back out and is focused onto the pinhole. So we say then in optical terms that this pinhole is conjugate with the plane of focus, the plane we're interested in. Now, if we look at what happens to out-of-focus light, the dashed line here, that light was in fact coming from further back and is focusing behind the pinhole, and then light with a very fine is also not focused on the pinhole. And what happens to that out-of-focus light is that it's blocked by the pinhole. So in a normal photo, you see the in-focus plane and you see all kinds of blurry structures which are out of focus. But in a confocal image, you see the in-focus plane and everything else is black because the light does not get through the pinhole from the other planes. And that's the big advantage of confocal imaging. It gives you the ability to do optical slicing, to actually choose a plane of focus and only look at that plane. Now, this image is, this schematic is drawn as if the optics were perfect, and so the confocality breaks down when you have poor optics, and I'll show you how that happens. So we have our confocal scanning laser ophthalmoscope. Ophthalmoscope just means that you're looking at structures within the eye. And the advantages are the increased illumination that you're allowed under the safety guidelines with a scanning beam over taking a flash lamp photo. The reduced effect of scattered light, because that scattered light doesn't get through the pinhole, and so that enhances your contrast and your resolution. And then you get further enhancement of the depth resolution, as I described, because you're only getting light from the plane you're interested in. And actually, if you can get a small enough pinhole, you also get enhanced lateral resolution. So this is a fundus photo of the back of the eye in a normal subject. And I'm not sure you can see it terribly well because the contrast isn't that high on the screen, but this is the optic nerve head where the nerve fibers are exiting the eye and the vessels, blood vessels are exiting the eye towards the brain. And you can get a sense of what a normal picture looks like. The subject is actually looking at this pointer. It's not really on their fundus. Maybe we should turn some of the lights down because I don't think this is quite as high contrast as I expected. My laptop's showing a better picture, and that's unusual for my laptop to do better than the projector. Okay, so this is the same subject in our confocal scanning laser ophthalmoscope, and so I want to highlight some differences, which I think even in this projection you can see. So the first is there's a slightly different magnification, obviously, but we can see more of the structures in the nerve head. On this image, you can see more of the pore structure because the contrast is increased. And also, where the vessels are exiting here, we're taking a cross-section of that vessel. And if we go back, you can see how you can see much more of that vessel in out-of-focus planes that you don't see here. You slice through the vessel, and these are actually slices, optical slices, through some of the other vessels. So that's a big advantage of this instrumentation. And this is another image taken in the lab, and there's many people in the audience who assisted with taking these particular images, actually, and so I say thank you to Chris, Liam, Marcia, Jennifer, and everyone else who assisted in these experiments. And this is a younger subject, again, an optic nerve head, and now you can see the structure of the lamina even more clearly in this image. We are interested in imaging the optic nerve head, I should say, in particular because changes in the optic nerve head are associated with glaucoma. We are interested in the three leading causes of blindness in North America, which are glaucoma, diabetic retinopathy, and age-related macular degeneration. And so the optic head is an important structure to look at in the case of glaucoma, as is the nerve fiber layer, which you can see some hint of here. And we are also interested in improving the images of blood vessels. Blood vessels do change in any kind of angiogenic disease, and so in diabetic retinopathy and age-related macular degeneration, we're interested in being able to image the blood vessels better. And those aren't the only applications of this technology, but they are certainly three of the most important applications. This is then a picture of the fovea of a young subject. And the fovea, this area here, is the area of high-resolution imaging or high-resolution vision. So you have many more receptors in this area, and so that's what you fixate with if you want to actually see something, and your periphery has much lower resolution. We also, of course, are interested in imaging in disease, and here we have images taken in early age-related macular degeneration, which is the leading cause of blindness in older adults. And so you can see here the abnormal drusen that are present in these two subjects. And this is just a reflection from some of the optics of the system. So I alluded to the fact that we can improve our cross-sectional resolution, and so I want to talk to you a little bit about why that's important. This is the retina, which is at the back of the eye, and these are the various cell types within the retina. And we want to be able to resolve in depth so that potentially we can separate the different cell types. The initial cell types that have been imaged using this technology are the cones. They are the photoreceptors, the cells that are light sensitive and phototransduce the light to an electrical signal. And they are waveguides, and that gives them quite high contrast in these images. The holy grail of this research at the moment is to image individual ganglion cells within the retina. And that can currently be done in animal models. There are some exciting new results backfilling living ganglion cells in animal models, including most recently the primate model, so that individual cells can be imaged. And that requires adaptive optics, which I will talk about in a little while. So we want to improve our depth resolution to be able to differentiate the different cell types, and we also want to improve our lateral resolution so that we can pick up changes through laterally in the retina. And here we have an example of depth resolution in a non-AO corrected system. So this is with at our reference plane for this particular image, and this is then imaging more posterior in the nerve head and more anterior. And what you can do with these kinds of images is then construct a three-dimensional picture of the optic nerve head. And so we want to improve the resolution of these three-dimensional pictures by using improvements to the optics of the eye. So that's where I want to go now in the talk, is to talk about taking a blurry image and fixing it, and how we go about doing that in our particular research. So when we take the light and it comes from the source and goes into the eye, we would like it to be perfectly imaging to a point at the back of the eye. And then after reflection out of the eye, we would like that light to be imaging to a perfect point when it goes through the pinhole in the confocal setup. And in order to do that, what we're going to do is we're going to put in an adaptive optics device, a deformable mirror. A mirror that we can change the shape of in order to correct the optics so that we get a very sharp point both here on the retina or the fundus and here after imaging through the confocal pinhole. And these deformations are of the order of fractions of microns that we have to adjust the shape of the mirrors by in order to do this correction. Okay, so to explain that in a little more detail, If we have an optical system that isn't perfect, then rays that go through the system will focus at different points. So here we have central rays that are actually focusing back here, and peripheral rays that are focusing here. And this is referred to as an optical aberration. And the particular optical aberration sketched here is a spherical aberration. And you can see then that you get a blurry point where you had a perfect point in the original object. We generalize this concept to describe wavefront aberrations. And a wavefront aberration is just the wavefront of light that's coming from the point. And so if we had a perfect point source producing a wavefront that was incident on this system, then after it's refracted through the optics, if it was still perfect, it would be a spherical wavefront, and we call that the reference sphere. And this that I've shown is a cross-section of that spherical wavefront. So that wavefront would then focus at this point, and geometrically it would give a perfect point. But the real wavefront, because of the optics not being perfect, is not distorted from that reference wavefront. And we are interested in exactly how large the difference is at each position in the pupil. And that's called the wavefront aberration. It's obviously a function of pupil position, and we have to measure it, and then we have to correct it. And you can see that the ray from out here is focusing at a different position than the ray from here, and so it's giving a blur on the image plane. Now, why is this so important? And it's fundamentally so important because every one of our subjects has a different amount of wavefront aberration. And furthermore, their wavefront aberrations vary continuously over time, with accommodation, from day to day, with pupil size. And so we have to dynamically track what their aberrations are doing. Okay, and how do we do that? Well, I kind of cut off my label there. We measure using a Hartmann-Shack device. Okay? And a Hartmann-Shack device is a device used by astronomers initially to measure the aberrations in the atmosphere. So the astronomers, if you look up Shack-Hartmann devices or Hartmann-Shack devices, you'll find some beautiful pictures of things that can be done by correcting the aberrations of the atmosphere to greatly improve images of stars. The big difference between an astronomer and myself is, one, I've got to put it into a smaller instrument. I don't have a huge telescope sitting on top of a mountain. And secondly, I don't have the money that the astronomers have. I don't have the millions of dollars to buy the mirrors that they can buy. And furthermore, I would like to see this technology out in clinical instruments. And optometrists and ophthalmologists don't have millions of dollars to spend on the instrument they're using. And so we have to get this technology miniaturized and cost-effective, as well as working to correct the aberrations of an eye. So that's the intent, and we're going to measure those aberrations using this technique. We put a ray into, or a wavefront into the eye. It's going to be diffusely scattered out, filling the pupil. And then in the return path, we're going to put the Hartmann-Shack device here, and we want to detect the shape of the wavefront, which is the white curve, and we're going to measure the difference of that wavefront from the black, planar, perfect wavefront. Okay, so it's an array of tiny lenslets, the Hartmann-Shack device, and if the wavefront is a plane, then each of these tiny lenslets focuses to a point, and I get a perfect grid of points in my image. But because the wavefront has a different shape, this little tilt here causes the spot to move, and this little tilt here causes the spot to move downwards, and so my spots move around, and I need to measure those spot positions relative to the ideal case. And this is a Hartmann-Shack pattern from a human eye from one of our subjects. And you can see that some of the spots are closer together and some of the spots are further apart, indicating the wavefront aberration across the pupil. And this is the pupil plane, and this is the extent of the pupil of this subject. And so we're going to measure those aberrations as a function of pupil position. And we can also then plot the aberrations in a false color plot. This is negative aberration, where the wavefront is behind the plane or wavefront that's expected. This is positive aberration, the wavefront is in front. And these units are microns of displacement of the wavefront from ideal. And you can see, in general, they're asymmetric. In general, they're bigger at the edges of the pupil, and that's a wavefront aberration for a human subject. This is the blur on the image plane that results from that wavefront aberration. And it also is asymmetric and blurry. For those of you who do know optics, the ideal blur would be an airy disk, a diffraction-limited airy disk, and this is much bigger and a different shape than that airy disk. Okay, so we come back to this adaptive optics element, and what we're going to do now then is we're going to measure how the optics would distort the wavefront. So we want, with the Hartmann-Shack, to measure the outcoming light and the distortion in the wavefront. And we want to pre-distort the mirror so that this pre-distortion plus the optics of the eye gives a perfect wavefront on the way in. And then on the way back out, the optics will distort the wavefront. And again, combined with the mirror, we will get a perfect wavefront focusing to the image plane. And so the amount of distortion you put onto the mirror is directly proportional to the amount you measure in the wavefront for that individual subject at that particular time. Okay, so we can correct the optics, and you saw in 2 dimensions here what the blur looks like. So we would go from this blur to a much tighter blur as we correct the optics for that particular point source. But perhaps of even more interest to us is what happens to the blur as a function of position along the axis. So here we have the blur produced by a point source running from anterior to posterior through the axis of the eye, and you can see the aberration in this particular eye has resulted in a double hump of intensity, and basically that's not very localized. We can't localize the light intensity. And after correcting with adaptive optics, we get a much better and much smaller half-width to that intensity, and now the energy is localized. And that will have 2 effects. It will improve the amount of optical slicing you can do. The resolution of your optical slicing will go up, because when you have this much blur, not all the light is going through your pinhole. It's getting blocked because of that aberration. And this will give you a sharper image, a brighter image, and an image with the ability to resolve axially. But much more exciting to our group is the implication to any kind of light-based therapy. So we are looking at exciting drugs with light in the treatment of eye disease. And in order to do that and make the treatment localized so that you only treat, say, a blood vessel, and you don't treat any other structures that are adjacent, then what you want to do is you want to be able to tightly focus the light so you can choose the structure you're going to actually treat. And so there are huge implications for light-based therapies of the ability to take the light distribution of a single point from this down to this. So when we correct our confocal scanning laser ophthalmoscope, We put in the Hartmann-Shack device to measure the aberrations. Then we put in the deformable mirror to do the correction. And they work in a continual loop to keep the eye corrected. And this is one of the advantages we have over the astronomers. The astronomers' loops have to work at kilohertz rates to keep the correction up for the atmosphere. Fortunately, eyes don't vary quite as rapidly as the atmosphere, so we're not working at the very high speeds that the astronomers are. But the effects are quite apparent. So these images taken in collaboration with Austin Rorda and our lab, you can see the photoreceptors, the individual cones, prior to correction of the optics, and then you can see a cone image after correction of the optics. And you can see that we can resolve the individual cones. They're the bright white structures. And we can also resolve here a small blood vessel running through those cones. So a huge improvement both in resolution and in contrast. So what kinds of deformable mirrors are we working on? We're working on the Genetics mirror, which is the mirror that's been used for astronomy for many years and is quite expensive, but works quite well. Its limitation is the amount of stroke. It doesn't have enough stroke to correct eyes with larger aberrations, and those tend to be diseased eyes. Diseased eyes have more aberration, and so you need a mirror that can correct a higher aberration. So there are some problems using Genetics besides its cost, and so currently other labs are working on MEMS mirrors, MEMS devices, which are much smaller and much cheaper. Again, as yet, they don't have quite the stroke necessary for the correction of the aberrations. We are working on some ferrofluidic mirrors as an alternative, which I want now to talk a little bit about. And this project has involved a number of people from my lab, Marcia Kiselak, James McPherson, and now Aidan Seaman, in measuring the characteristics of a ferrofluidic mirror. So I should describe that to you a little bit. The ferrofluid has nanoparticles which are magnetic, and they are suspended in a liquid. And then you float on that a metallic liquid coating. And basically, after that, you can apply through your individual actuators a magnetic field, which will change the shape of the surface of the mirror. So I'll return a little bit later to, if I have time, to the ferrofluidic mirror, but meanwhile I want to show you the other advantage of the confocal scanning laser technology, which is that we end up with, that was going to loop, my apologies. We end up with video rate images at 30 frames a second, and in these images, in fact, you can see blood flow. That's what you're seeing. There are leukocytes going through the vessel, and this then allows you to track what's actually happening on the retina. Okay, so this was an adaptive optics corrected image of human cones, and I want to show you now an image, an early stage image from our latest work, and it's perhaps not going to show up terribly well with this projection, but I'll describe it to you before I run the movie. These are reflections, and so they're going to move, because what we have done is we have processed these images to keep the photoreceptors, which are the little bright spots, in a constant position during the movie. So we have actually taken all the frames and superimposed them so that the position of the individual photoreceptors doesn't change because of eye movements. So if I go back and rerun this, should have mentioned eye movements. So the movement you see there is due to the eye movement. You can see the blood flow, but you can also see the whole image moving. And so what we have done is used software that's available from the Rochester group to actually compensate for the eye movement. So the images of the individual receptors here are going to stay fixed. You'll see movement of the reflections, but the photoreceptors will be relatively fixed. This image is non-AO-corrected at this stage, but it is the first image of the living cones at the back of a chick eye, and so that's why I want to show it, because it's an exciting new result from work with Marcia Kiselak and Beth Irving. And if you focus on one of those bright spots, you'll see that the bright spot, although it scintillates, moves very little, the small spots, during that movie. And this is using, as I say, software that's available through other groups, and it's very exciting that from these kinds of images you can also track eye movements of your subjects. The photoreceptors at the back of a chick eye are particularly important because they will change with growth. The chick is an important model of eye development, and also they will change with the induction of myopia potentially. And so we are looking, we're beginning to look at the spacing of cones during the induction of myopia and what's actually happening on the retina with relation to the optics during myopia development. Next, I want to talk about some of the work we're doing currently on age-related macular degeneration and the potential of two-photon therapies to treat age-related macular degeneration. This project is, in fact, a national project. It's a Center for Photonics Innovation project, and so we are just one center in this project. Our expertise is in the optics and in correcting the optics so as to localize the light delivery at the back of the eye. So for those of you who don't know, age-related macular degeneration in its wet form, in its neovascular form, involves proliferation of blood vessels which then come up through Bruch's membrane and into the anterior part of the retina, and this is, as I say, a leading cause of blindness in older adults. And so what we want to do is potentially be able to treat these vessels before they leak and do more damage to the surrounding tissues. And so, as I say, we've done some imaging in early ARMD, but the project I want to talk about primarily is the photodynamic therapy with two-photon excitation. So photodynamic therapy, Visudyne, is a drug developed by QLT in Canada that is an approved treatment for AMD. And it's done just with a flood illumination excitation of the drug. Two-photon excitation and imaging has the advantages that we can localize the excitation of the drug, and we can use, instead of the longer red wavelength that's used to excite the drug, we can use infrared light for drug excitation, which will penetrate deeper into the fundus and allow us to treat potentially into the choroid. There's less scattering of this infrared light, and there are higher damage thresholds for adjacent tissue if you're using infrared light rather than visible wavelengths. So there are lots of advantages to using two-photon excitation. But the biggest one that I want to emphasize is the localization of the excitation. So going back to this plot of what a point source looks like on the retina without adaptive optics correction, it's quite blurred. And with adaptive optics correction, it's quite narrow. And the excitation of the drug with a two-photon excitation is proportional to this intensity squared. And what that means is that this will narrow even further when you square this function, and it will allow you to do your excitation in the axial dimension much more localized than without adaptive optics, and much more localized than in the single photon excitation. So single photon excitation goes as intensity, and here's the slide that I'm sort of alluded to. So single photon excitation goes as intensity, and so you'll tend to excite all along this beam, and two photon excitation goes as intensity squared, so you will localize where you are exciting the drug. And so that's why this methodology is so important. We are beginning these experiments in rats, and the reason for that is because the small rat eye actually has even a higher lateral resolution than the human eye, which is going to have a 3 times lateral resolution and a 10 times depth resolution. So we will be able to track much more easily in this animal model what we're actually doing when we attempt a 2-photon excitation. Not to mention the fact that we couldn't just go out and do the therapy in people right away. We have to prove it in an animal model. So we have been measuring the aberrations in the rat eye in awake rats, and that's a Hartmann-Shack pattern from an awake rat to characterize these aberrations. And in the very near future, we're hoping to actually correct the aberrations. We've also done some initial experiments choosing the pupil sizes that we want to use for these experiments. And also there is one of the CSLOs that we designed is actually at Princess Margaret Hospital, and the initial 2-photon experiments have been performed at Princess Margaret. So these are CSLO images from a rat eye taken in our apparatus, and you can see that we're resolving the blood vessels quite well. And these images were taken as part of Mark Bird's thesis to look at the best optics that we could achieve in the rat without the adaptive optics correction. And the conclusion from that is that the adaptive optics correction will definitely be necessary to achieve the localization and resolution of imaging that we want. Okay, so I'm rapidly running out of time. How many people here have a specialization in optics? I know Vengu does. Anybody else? Nobody else who would say they were, well, and Chris will be interested in this, of course. So I'm going to talk just very briefly then about Mueller matrix polarimetry. By putting in particular polarization of light and then measuring a particular polarization of light, we can further improve the images of the fundus. And we do this by knowing that we are only looking at the intensity of the light. So for those of you who know polarization theory, this is the complete Stokes vector, but we're only interested in the intensity. And we look at the various images produced in different polarization states, and then we recreate images across the Poincare sphere to maximize various image quality metrics of the images. And we can show greatly improved imaging. And one of the metrics that we've often used is contrast across a vessel. And those of you in vision science know the definition of Michelson contrast. And we can show improved contrast with this method by comparing the original images and the best images that we reconstruct. This has led us to a subject area that may be of more interest to some of the people in the audience, and that is the area of image quality metrics. And this is an important area both for looking at the optical quality of an eye and whether that quality has improved, and also for looking at whether images are actually better. And so this was the focus of Jennifer Hunter's PhD thesis, and she's done quite a bit of work on image quality metrics and which ones you should use when you're trying to get the best possible fundus images. And interestingly, doing different things with different metrics actually produces, in combination with the polarimetry method, produces different views of the fundus. And so you can see that these images are quite different. This one emphasizes small details here, where this image emphasizes the neuroretinal rim. And this is a very exciting area for us because we're going to get much more additional information by manipulating the polarization properties of light. Okay, so when we actually get to clinical applications, another thing that we've done is to modulate the laser beam in our system using diode lasers initially. And that's going to be important both to visual testing, so we can write on the retina, and this has, of course, previously been done as well. But the direct laser modulation has the potential to apply the retinal writing to therapies as well as to vision testing. Okay, so I'm going to wrap up now, and then you can all go eat, and your stomachs will stop rumbling. This work is very important to the diagnosis of various diseases, and I touched on glaucoma, AMD, and I mentioned diabetic retinopathy. Photoreceptor diseases are also important and can be easily detected using these methods. And most important, in the animal models, we can potentially track treatments, experimental treatments for many of these diseases, including the retinal degenerations, to see if the therapies are actually treating the disease. And so the tracking of therapies in the animal models, in the rat and mouse models in particular, is a current and future area of research in the lab. It's very exciting if you can use the same animal rather than having to use many more animals and not track an individual animal. And the in vivo imaging allows you that individualized tracking. We obviously are continuing with the myopia research, and this instrumentation is important to the study of normal and abnormal visual development in humans and animals. And obviously, I've shown you some improved images of structure and some the potential to develop novel light-based therapies. I didn't dwell very long on the actual functional testing that can be done with these instruments, but if you can localize the light, then you can localize your visual targets, and you can actually do much finer functional testing as well with this kind of instrumentation. So in our lab in particular, our focus in the near future will be on better delivery of high light flux in a localized volume. That's particular to the 2-photon project. Continuing with high contrast and high resolution imaging for a number of our different projects. Developing these instruments for specific applications. And one specific application is for animal models and imaging of rodent eyes. The other specific application that we will be developing an instrument for is the imaging of children's eyes, and this will be in collaboration with Sick Kids Hospital. We are also collaborating with Kostas Besheva on adaptive optics correction of a combined optical coherence tomography CSLO instrument. The Sick Kids project will concentrate in the first instance on diabetic retinopathy from type 1 diabetes, and we will continue to develop the high-resolution imaging of myopia development. My support comes from a number of sources. CIPI is the Center for Photonics Innovation. It's the National Photonics NCE. I also get funding from the Center for Photonics, which is an Ontario center. And NSERC, they recently awarded CFI, awarded jointly to Sick Kids and the University of Waterloo, and also some industrial support. So that finishes my presentation. If there are any questions.
To ask questions, I would just ask you to be patient so we get the microphone to you. Make comments or ask questions. I'll start with see if I see any hands. If not, I've got a ton of them. So by the way, we allow dumb questions at Monkey's very fine.
I'm just going to grab actually some orange juice.
That's why I'm allowed to be here, so it's just the dumb questions. Why don't you talk at the beginning about an interest in transforming this work into an instrument?
Yes.
I wonder if you could comment on that at all. Like, where are you in that? What are your hopes?
Right. Well, I should have said that all of the images I showed you were actually taken with our instruments, so we do have an operational instrument in the lab. Our hopes, as I was saying, are to redesign the instrument with the two specific focuses, and the CFI recently awarded is going to help with that. One is for infant and children's vision studies, and the other is for the rodent studies. So we're hoping, in fact, to build two new instruments with those two focuses.
What about your orientation to creating something that can be used in the average office? In other words, I'll call it an industrial distributable interface.
Right. The adaptive optics will be miniaturized, and the first smaller instruments are being produced. I think it's a little soon to see the technology going out into the individual office. I think the next approach is to place the instruments in hospitals, where they can be used by a number of clinicians and more efficiently used. But yes, obviously, we hold certain patents, particularly in the polarization imaging, and we would really like to see the instrumentation get out into the clinic.
So you're working with companies with regard to this?
Yes, and having ongoing discussions with companies, some of which I mentioned and others I didn't. So, yeah. And we're happy to talk to any other companies who might be interested in this technology.
Questions or comments from you? Here's one here.
The polarization, in the polarization work, are there any specific polarization states that you find to be optimal?
It's interesting. What we found is that the specific polarization state that is optimal depends on the subject and on the structure. So we started off with a global metric and saying, okay, we're going to maximize this metric, and we looked. What I can say is that circular polarization is important, that linear polarization alone is not enough, which is, of course, what a number of the commercial instruments currently use. So that would be my first comment. The second comment is that we have found that it depends on the structure you're looking at and the particular subject.
Well, part of the reason I asked is I'm thinking of these photon generators, and where you could get specific forms of the Stokes or Miller matrices for the photons that are generated, and maybe you could fine-tune those. That's one. And then secondly, you could also, for correcting for the aberrations, instead of doing micromirrors and all that, I'm wondering if there is a way, I'm just speaking out, of using retardation plates to compensate for some of this to get better contrast.
Yes, it has actually been shown that you can use phase plates to correct some of the aberrations, and so Steve Burns has shown that. The difficulty with the phase plates is their alignment has to be very exact. That's the first thing. And secondly, of course, you'd have to produce a different phase plate for each individual subject, except that you can use a Hamamatsu LCD device where you actually place the phase on the device for each individual subject. Their stroke is an issue, that those Hamamatsu liquid crystal devices don't have the kind of stroke that we're looking for. We're looking at relatively aberrated optical systems when we look at eyes. In answer to the polarization question, I didn't do much detail on how we do the reconstruction, but we basically take images with 4 different polarization states as our base images, which we then use to explore the Poincaré sphere.
At the end, you mentioned the word tomography. Can you explain that a little bit? I mean, are you scanning down, for example, down the rods and then reconstructing them? Is that the idea of that? Or?
The idea is that this instrumentation is very powerful when it comes to tomography. That's not the primary focus of our lab, so our attempt to localize the light in depth is aimed more at the treatment angles. The tomography is also done by optical coherence tomography. optical coherence tomography, and it actually has higher axial resolution than the confocal scanning laser ophthalmoscope. So if you're just imaging and you want the best possible tomographic reconstruction, then you would probably go with OCT, the ultra-high resolution OCT, which is the area that Kostatinka Besheva is an expert in. But if you want to actually deliver the light for therapy, then you're going to use the instrumentation that we have, and that's why we're exploring collaborations with her doing a combined instrument.
Now, with the technology, let's say they're doing it to kids, I don't know how far you're going with this, but can you actually reconstruct a sort of a three-dimensional view of the retina?
Yes.
And how deeply can you go? Can you get to the ganglia?
Oh, absolutely.
Okay.
So I, in fact, had one. I can just go back to it. It's probably faster to just flip back till I get there. There's one. So this is a reconstruction of this nerve head going from the surface up here down into the very deepest layers. So this is a mid-range picture, and we've got pictures in front and behind that one. And then this is the surface of maximum reflectivity plotted as a function of position in depth. And so this is microns, but we're going 2,000 microns, which means that's 2 millimeters deep.
Interesting.
So, yeah.
So can you actually get anatomical reconstructions, in other words, where you see the physical structure as opposed to a
Yes, and you can rotate them, and a number of labs are concentrating on that aspect of the technology. That's not the area that we've concentrated on.
All right, two final dumb questions. One, to what degree does the cornea contribute to the eye's function as a focusing instrument compared to the lens and...
In terms of power, about 60% of the power of the eye is from the cornea in a human. In a rodent, it's more like 20%. So the crystalline lens is much more important in the rodent eye than in the human eye in terms of power. In terms of aberrations, they both contribute to the optical
Now, in your
aberrations.
Hartman Slack, you showed the array of points. At one of the pictures, you had down in the bottom right corner a couple of bright spots. Is the brightness of any interest?
I should learn not to use that picture. Everyone always asks
Okay,
about
just
brightness.
so it's a great dumb question.
We assumed that was a tear film effect because, of course, and the aberrations are also contributed to by the tear film, and we assumed that was just some bright area of the tear film that gave that brightness. Mm-hmm.
Other final questions or comments from anyone? If not, I wanted to thank you. First of all, very interesting. I learned so much here. It's incredible, more than I ever knew before about it.
I hope you remember it afterwards.
Oh, of course, of course. And the other thing I was thinking along the way, at NSERC, they're always asking people to have focus in their research. You must have never had a problem with that doing your grants.
No, I have the optical focus in my research.
Thanks very much.
Thank you.